HIV is a virus that infects millions of people around the globe each and every day, making them very, very sick. Over the years, the number of infected people has continued to grow, and although we have medications to treat the disease, they are very expensive, leaving these sick people with a very large financial burden. HIV is a virus that has an envelope made of the same molecules as the plasma membrane. Inside the envelope, there's a capsid made of GAG proteins, and the capsid works to protect the virus's genome. The genome is made of 2 strands of RNA, and all over the surface of the virus there are envelope proteins. HIV infects T helper cells. These cells are our body's soldiers that fight against foreign invaders like bacteria. On the plasma membrane of T helper cells, we have proteins that act like locks, and the envelope proteins on HIV work like keys. When HIV puts its key into the lock, it is able to fuse with the plasma membrane and enter the cell. Once it fuses, the capsid moves into the cytoplasm and comes apart. The RNA is changed to DNA, the same molecule that carries all of your genetic information. The new viral DNA then moves into the nucleus and becomes a part of the cell's DNA. There, it can remain for a very long time. When it decides to make new viruses, the DNA is once again changed to RNA and moves out of the nucleus. That RNA uses the cell's ribosomes to make its own proteins. Ribosomes in the cytoplasm make the GAG protein, while ribosomes in the rough ER make the envelope proteins. The envelope proteins then move through the Golgi apparatus, and vesicles from the Golgi help move the envelope protein to the plasma membrane. The GAG proteins and 2 copies of the virus's RNA also move to the plasma membrane. Once they're all there, the virus is able to bud off and move on to infect new cells. Imagine going to the amusement park with your brother and your mom. You all go separate ways, and when it's time to leave, you're all scattering around to find each other. Wouldn't it be more efficient if you found each other before it was time to leave? Then, when the park is closing, exiting would be much faster and much easier. Well, for that same logic, some scientists think that the GAG protein grab a hold of the RNA and attach to the envelope protein within the vesicles before meeting at the plasma membrane to exit the cell. Once they meet up inside the cell, all 3 components move as a unit to the plasma membrane where they can bud off to form an independent virus. I work in the lab to try and figure out exactly how the new viruses are made. If we know how they are made, then we can hopefully design a new medication that will come and stop the virus from being built. Hopefully, this new medication is cheaper and more effective than the ones we use now.